Hi everyone, welcome to my channel. I'm Taposhi and today I come with another video that is obviously you can see the cytokinin. This is the plant hormone, and before going to this topic I want to say that thanks to all of you who have already subscribed my channel and support me for the growth of my channel. Okay, and obviously I explained different plant hormones that in my previous videos. You just check out and continue with me and my channel. Okay, let's start the cytokinin. What is cytokinin? Cytokinin is actually the N6 substituted adenine. Okay, adenine derivatives. These are actually nitrogen 6 substituted adenine derivatives. And at first it was isolated from the corn and it was called the zeatin. Okay, it was called zeatin. And during the infection of some pathogen, you can say here, this is one kind of bacteria. Its name is A. tumefaciens. That when it infects the plant cell, it's you know all that the bacterial DNA that incorporated with the chromosomal DNA of the plant or the host body. Okay, and the important thing is that plasmid. What is plasmid? Plasmid actually the bacterial a small portion of Ti plasmid here, obviously small portion of the Ti plasmid that is called the tDNA. Okay, this is called the tDNA, and this is very important for the biosynthesis of the cytokinin. Okay, if I think one second. So here actually the virulent strain is the agrobacterium, and what is called the plasmid? The circular DNA that present in the bacterial cell. This is called the plasmid. Okay, and the very small portion of the Ti plasmid, that is called the tDNA, that is very important for the biosynthesis of the cytokinin, because this tDNA that carries gene, one kind of gene that is very important or that is necessary for the biosynthesis of the cytokinin. So what is we learned here? This is the adenine derivative, and this is first isolated from the corn and known as the zeatin. And infection of the A. tumefaciens, that is a plant. What happens? When it infects the plant's body, it's DNA that incorporated into the host cell, and this is a virulent strain, another that is called the agrobacterium. Okay, and the plasmid is called, what is called the plasmid? The plasmid is called the circular DNA that present in the bacteria, and the Ti plasmid, that is called the tDNA, very important, and it is obviously from the A. tumefaciens, not the it's agrobacterium. In case of agrobacterium, the biosynthesis of the cytokinin in another way is possible. Okay, but in case of this is the Ti plasmid or the tDNA that enter in the plant's body from the bacteria, this one, its biosynthesis is possible in such a way. Okay, and here the gene that is very important or necessary for the biosynthesis of the cytokinin. That's why the tDNA or Ti plasmid very important for this cytokinin. Okay, so now we learn the biosynthesis process, what is happening here. You can see here the IPT. What is called the IPT? IPT is the isopentenyl transferase. Isopentenyl transferase is the enzyme, and DMAPP, what is that? This is the dimethyl allyl diphosphate. This dimethyl allyl diphosphate, when added with this ATP or ADP, then in presence of IPT enzyme, it converts into TPRTP or IPRDP, and this conversion is not stable. This product, after production of this substance, this convert into ZTP or ZDP, and in presence of enzyme this enzyme is called the cytochrome P450 monooxygenase. Okay, cytochrome 450 monooxygenase. So this after the formation of the ZTP or the ZDP, that is called the zeatin, okay, ZTP or ZDP, that is called the zeatin, that converts into trans-zeatin riboside 5' monophosphate. Trans-zeatin riboside 5' monophosphate, and this converts into trans-zeatin riboside. Okay, this is the actual structure of this, and this is also not stable. It also converts into trans-zeatin. This trans-zeatin is very important. Okay, this trans-zeatin is actually the stable substance. Okay, this trans-zeatin, and this is very important, and you already know that actual cytokinin that was first isolated from the corn, it was known as the zeatin. So this is the actual product of the cytokinin. Okay, so this is the biosynthesis of the cytokinin in this way. Another way that is possible when the agrobacterium is present, in that situation how the cytokinin biosynthesis is possible, I just explained very briefly. Okay. In case of agrobacterium, this is the agrobacterium. What happened here? Here, this is HMBDP. HMBDP full form is the 1-hydroxy-2-methyl-2-E-butynyl 4-diphosphate. And this here, in presence of IPT enzyme, IPT isopentenyl transferase, it added with AMP. Okay, it added with AMP and transform or convert to the ZMP, zeatin monophosphate. Here we see that first it is from the ZTP or ZDP, but here it is from the ZMP. Okay, because at ATP or ADP is not added here, AMP is added, and here the another substance, here you can see the DMAPP is the main substance, but here is the substance is the HMBDP. Okay, so from here HMBDP, in presence of this enzyme, AMP added to react to form the ZMP. Okay, and this ZMP transform or convert to the TZRMP, that is the trans-zeatin riboside 5' monophosphate. Okay, so from ZMP this is transformed here, or this is the conversion of the ZRMP is possible, and here ZTP or ZDP is important, because from ZDP it is formed the trans-zeatin riboside monophosphate 5' obviously. So these actually two difference, this is seen because this is virulent agrobacterium, so here this situation is occurring. So this is the actual the biosynthesis of the cytokinin. I think you can understand it properly, that what is the how the biosynthesis of the cytokinin is possible, and here obviously tDRNA has important, you can say that it is important factor. Okay, so we learned here the biosynthesis. Now we learn here that the functions of the cytokinin. Okay, the functions of the cytokinin. Okay, so now we learn about the function of the cytokinin. So now we learn about the function of the cytokinin. So here you can see the first one is the cell division, second one is the synthesis, and the third one is the chloroplast saturation. So in case of cell division, the positive regulator, cytokinin is the positive regulator of shoot formation or shoot apical meristem, and negative regulator of the root apical meristem. What is the meaning of that? That means it helps in the growth or the development of the shoot apical meristem, but it never help in the growth and the development, or it is not a good regulator. You can say obviously negative regulator for the root apical meristem. And here I mentioned another word, the auxin and the cytokinin both are related to each other. That means the ratio of the auxin and cytokinin is very important for this root growth and the development of the root apical meristem and the shoot apical meristem. If the auxin and the cytokinin ratio is high, that means the root formation occurring. Okay, and the next one, in the cytokinin and auxin ratio is high, that means the shoot formation. Okay, so that means cytokinin generally help in the shoot formation and the auxin generally help in the root formation. Okay, that's clear. Next one is the senescence. What is called the senescence? Senescence is the actually it is a process. What type of process is that? It is the energy dependent, genetically programmed aging process and which leads to the organ death. Okay, that means totally it is programmed in the plant's body that some genes are very important for their progression towards the death. It is totally programmed in their body, and this process, and obviously it is energy related, and this process is called the senescence. But some hormones that is present in the plant's body, sometimes they actually inhibited the senescence process, or they delay the senescence process, and sometimes they accelerate this process. Okay, so cytokinin is the negative regulator. That means it try to delay the senescence process or its progression towards death. Clear? And what happened in that situation? As the plant tissues senescence, they produce enzymes necessary to recycle the materials and reroute the subunits to areas for use by active growth hormones. Okay, so here what is saying that plant tissue that already progression in the senescence, they actually produce some enzymes. So these enzymes, why it is necessary? Because these are necessary to recycle the material and reroute the subunits to areas for use. Okay, so again they try to recycling the materials for using that in a proper way to grow for their growth and the development. Okay, and here this hormone, this cytokinin, it has more actively it perform or it plays a role to mobilize the nutrients. So you can see, or you can say that if the nutrients energy is totally used in a part of the body, that means so there the programmed cell death must be possible. So what happened, or what is the actual role of this cytokinin? It try to mobilize the nutrients in such a way that the particular area they can get the nutrients for their growth and the development. As usually, or that's why the senescence process or programmed cell death that may be delayed, okay, in that part of the plant body. This is actually the role of the cytokinin. Okay, and the third one, that is the chloroplast maturation. In sometimes the cytokinins that promote the conversion of the etioplast. What is called the first we know etioplast. Etioplast is the dark grown seedling. Okay, it's dark grown seedling, and here the protochlorophyll is present, not chlorophyll is present. Okay, and that due to the presence of this protochlorophyll, they do not synthesize any enzyme or the structural proteins to synthesize the chloroplast. So here the plant's body is not looking like the green. In that situation, the cytokinins that have important role, they transform the protoplastics. Actually, etioplasts, these are formed from the protoplastics. And what is the role of the cytokinin? This cytokinin that converts the etioplast into the chloroplast. Okay, etioplast into the chloroplast. And what is that? Via the stimulation of the chlorophyll synthesis. Okay, via the stimulation of the chlorophyll synthesis. Okay, so this is the actually role of the cytokinin. Today we learn, and in my next video I try to explain the secondary metabolites part that actually remain. Okay, so I will try to describe that part that is related to secondary metabolite in my next video. So thank you. Please continue with my channel and obviously support me.